Okay, so welcome everyone to our 13th session of the Berlin Tokyo Viral Cloud. So my name is Georg Drömmel. I'm an artist. I'm a co-founder of the BioClub Tokyo, and together with Christian Delutz and Regine Rapp, we started this kind of conversation, these sessions between Berlin and Tokyo, actually a year ago, right? So or nearly a year ago, and as a way to build bridges and to keep in touch. So today we have two guests. One is Hideo Iwasaki from metaPhorest, and the second one is Matthias Rillig. So both guests will have about 30 minutes. And after this, we will have short, maybe 5 minute Q&A session. And after the two presentations, we will have a longer discussion. So let's start with Hideo. I think you can already
Okay.
start screen sharing. I will introduce you a little
Okay.
bit. So Hideo Iwasaki is professor at Waseda University. I actually forgot how your lab is called.
It's okay.
Hideo Iwasaki lab. But Hideo has a background in chronobiology. He was very instrumental in finding the circadian rhythm of cyanobacteria. So his lab concentrates on this field. But it's also very special because since more than 10 years, he opened his labs to artists, including me, who actually can use this amazing resource. And also a part of this is the metaPhorest biomedia, art and biomedia platform, we call it, right? And of course, Hideo is also not only scientist, he's also an artist. I think since you're longer an artist than you're a scientist, because I think I'm sure you will you'll talk about this. You started very early with this paper cutting kamikiri, where we now um, see very nice example. Yeah, and I think this. I don't want to take too much of your time. Okay, please, Hideo.
Thank you very much for your kind introduction. Um, okay. Thanks again, Christian and Regine and George, for inviting me today. And as many participants know, I gave a talk in this viral cloud meeting in last November, and so many audience were there. So to avoid overlapping, I will only, the last time I mainly talked on my own artworks. And so to avoid overlapping, and I will only mention briefly about my works, but instead I would like to talk much more about the metaPhorest platform, um, according to the George's request. And so my lab is basically a microbiology lab as Dr. Rillig, and probably a little bit different focus, but it's open to artists and designers, as George already introduced, and who are interested in biology and biotechnology or philosophy of life, and etc. So, and although my lab is very small, probably compared with the Dr. Rillig's big lab, but and so our attempt is to do the biological science and the biological art and some humanities studies related with life simultaneously in the same place, and so and it forms like some small society for biomedia art or bio-related art in Japan, and for some opinion exchange or information exchange and related with bioaesthetics or bio-related arts. And so and for metaPhorest information, we have a Facebook page, and if you are interested in, please check the, this page, as we announce some seminars or something like some activities on Facebook usually. And so additionally, it's important to note, and I do not ask artists or designers to decorate scientific activities. And at least in Japan, I don't know in Germany, but and in the context of science communication, and some artists and designers have been employed with in some institutions or universities with scientific budget and to produce good-looking illustrations or good-looking movies to sexy up scientific activities and to the public. But I do not deny this type of activity at all, but how can I say, it's not my cup of tea. And so I prefer coexistence of artist-driven project and scientist-driven project at the same time. And so 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 I founded it in 2007 and 14 years ago, and 24 members have joined our platform, and currently about 10 members shown here are pursuing their art projects. And first of all, I have to confess that our platform is not very well organized and compared with other pioneering platforms like Symbiotica or Hackteria. But based on a very fragile personal connections, and uh, and and the main reason for that is primarily due to my lack of ability of my administrative ability to manage organization. But generally, as our platform recommends the participants to make relatively longer partnership. And so this is the diagram of our membership and the 24 members here, and George is here. And it's debatable when George actually starts to join this platform. So, but anyway, officially, and like that. And so we we do not. I think that I do not ask artists so many demanding one, but so we asked artists to present their project proposal to science students when it's beginning. And also we asked the annual or regular report at metaPhorest seminars, and we have some bioaesthetics or bioart introductory course to the science students, and which is to make the risk, how can I say, project plan of their own bioartworks or something like that. And in such a occasion, we ask the artist to join it as a tutors. And and if and we do not demand them to use the lab equipment, but and but and in if they want to use the lab equipment, of course, and we they need to take some lab training course. And so, and also it should be noted that our lab has usually about 20 undergraduate and graduate students, or one or two postdocs who are dedicated to biological research purely. And so, but compared with that, the majority of the science students, the only one or two students and some artists like George are involved in art and humanities studies. So metaPhorest members are always minority in my lab. And even though the density of artists should be much higher for the standard scientific labs. And okay. And so there are several, how can I say, background to found the metaPhorest platform, but the one reason to make metaPhorest is to make a client bottle to reconsider liveness. And of course, main issue in biology is to ask what life is. But as I, so this is overlapping with my last talk, but life can be considered with at least two ways. And one is scientific target and being described as a biological complex or organisms. And of course, even in natural science, so we have a lot of the quite different approach to life, and it depends on the disciplines. And the chemists are want to understand the physical how the physical aggregate constitute the liveness, and also the maybe mathematicians want to know the equation of life, or we biologists use some biological notions like heredity or evolution or metabolism to explain the liveness. But anyway, the common feature is liveness resides in the subject in the species or some biological phenomenon or biological complexity or something like that. But in but the other is life interpreted as intersubjective or cultural presence, which is perceived to an observer as a more cultural or emotional object or relationship as life with us, and which is, in my opinion, highlighted in funerals or commemoration, for example, and it's very cultural and social or some personal. And I am interested in the ambiguous border of the two, the complicated relationship between the two subject. And to me, it looks like a client bottle. And so, and they are not separated, and they are tightly interconnected and form a very complicated boundary. And to explore this complex, I think that the art aesthetics and art is a very good platform, as is philosophy and ethics. And, so scientifically, the most of our lab members or science members are studying on the special temporal dynamics using cyanobacteria as model, combining genetics, molecular biology and biochemistry, genomics, microscopic observation, and mathematical modeling or something like that. And it's much simpler than the Dr. Rillig's the more complicated approach. But anyway, but interest to circuit and rhythms is not natural, and but very epistemological, inspired by some science studies, and that is the history and the philosophy of biology. So, studying on biological clock personally is not driven by natural scientific interest, but the already the some epistemological background from the beginning to me, and so it also make so the to me the scientific study on the biological clock in cyanobacteria is already living in the client pod. And also the colony pattern formation just look like worms, but actually they are the collective behavior of cyanobacteria. And and it's officially an expansion of studying temporal dynamics in chronobiology to that of spatial dynamics scientifically. But in reality, I started this project as a byproduct of my own biological art making project. So again, it also reflects some client bottle. Okay. And I'd like to talk about some examples from the metaPhorest activities. And usually I talk more on about the project with established artists like George, But today I will talk on the former students and postdocs who were metaPhorest members and who are the Juan Castro and who was a postdoc in our lab with art educational background, and Tomoyo Ishibas, who used to be a grad student in my lab, and also the more recent graduate student, Shiru Kirie. And Juan Castro is from Colombia and studied a graduate school at the Graduate School of Tama Art University under supervision of renowned artist Akihiro Kubota for media arts. And during his PhD, we started collaboration to make an interface between cyanobacteria and humans, and where the brain wave from the audience was converted into light information, which affects the movement of cyanobacteria. And so we made a live installation and combining the biomedia and sound and visual coding. And and then he moved to officially to my lab as a postdoc. At that time, I had started some interdisciplinary project on artificial cells, which I may talk you a little bit later. But and Juan was also fascinated by the complicated nonlinear dynamics of the lipid behavior in a test tube, and which is used as our synthetic cell membranes. So he got connected with some biophysicists and started a very mutual collaboration with them. And after about 5 years research, he published a very interesting paper, a very professional chemical research article in which he developed the new lipid molecules, which show very novel behaviors in vitro. So I think it's very rare case that the art background students became a professional chemist and to write the very professional papers. But anyway, so using this, now he is developing his own some chemical art related with boundary between the life and matter. And he's now associate professor at IAMAS at the Institute of Advanced Media Arts in Japan. And again, and yes, and another student is Tomoya Ishibashi, and who was a graduate student in my lab. And from the beginning, he was very fascinated by biomedia art approach after watching Oran Kat's show in 2009 in Tokyo at the Mori Art Museum, where I helped Oran to set up the victimless leather work. And so we explore some possibilities, and he finally did the hybrid biological artwork related with goldfish. And the goldfish are purely produced for visual amusement, and it has about 1,700 years history, and they are no longer suited for living in the wild because it had a fancy shape, and they swim very slowly, and it has a very outstanding color, so they cannot survive in the wild. And based on the crossing hybrid, he made the goldfish to regain the ancestral crucian carp-like fish, and like this. And so it was also done some genetic studies, and which was not done in science, and to check whether this type of the descendant has a, how can I say, a higher swimming ability, and he did some behavioral analysis or something like that. And you know, we are at the Department of Science and Technology, and so therefore, you know, to graduate it, we need to do some science scientific studies, and so um, like that, and then he made some installations using this one, and he now um, and he expanded his interest not only bio but also the more general media art and fine art issues. So, and I'm happy that he is a very active media artist right now. And and the third example is Shiryu. And Shiryu was a former graduate student and was highly interested in the cultural history of botany or horticulture. And also he is enthusiastic about the mathematical approach to morphology. And so, and he studied in investigation of the history of floriculture using a water lily as a model. And his scientific approach uh, was to quantitatively measure three-dimensional structure of flower cultivars and made a morphological diagram or to do some mathematical modeling and to investigate some genomic sequences to investigate the genomic sequences to map the morphological characteristics and so on. And he focused on the cultivars produced in nursery in France, which provided Claude Monet with newly produced cultivars. So since most cross-hybrid information in the Monet's era, Monet's time is lost. So in collaboration with the French flower company, he is now reconstructing family tree related with Monet's water lily. So based on research, and he's now starting to show his own artworks related with with this subject. And so this is the typical example of our science, the biological art activities, uh, if they were the students or postdocs here. And as a reference, and this brief introduction of my own one, and so this is my example to of how can I, to make some client support related with my own biological scientific activities with biogenic deconstruction, you know. So in this work, and the cyanobacteria are cultured on cutout of biological papers, which had been set on agar plates, and the complex patterns emerge while bacteria grow and move. And the scientific articles to be cut out are product of my own biological studies on biological clock. And so, and so and for details, and please check my website or the YouTube movie. Okay. And so this is another example. We set up two stone monuments for the souls of artificial cells and microorganisms in a public space near Fukushima. And this is to connect one of the most advanced and challenging biological attempts to make an artificial cells and one of the most typical intersubjective or cultural ritual related with life, that is commemoration or memorial service. And again, oops, for details, please check my website, and details were documented. And this is the scene of rituals, and yeah, some interviews to the synthetic biologists to discuss about what is liveness to or death to the artificial life or something like that. And yeah, and um, so so anyway, so synthetic biologists try to reconstitute scientific characteristics of liveness which resides in the object, but it finally demands us to reconsider if something synthesized is really living, uh, which turned to be a very cultural issue suddenly. So by setting this monument and holding memorial service leads to consider what life and death to means to synthesized life, and how this technology changed our view of life and death and so on, was the aim of this project. And for details, anyway, please visit my website. And so here are some titles of our recent humanity or aesthetics related themes from our lab. As, as I said, most of our students are doing standard microbiological studies in bacteria. And so these activities are less than 10% of our lab activities. But personally, I myself take time for science and art at maybe 1-to-1 ratio. But anyway, and it's not very, how can I say, so-called efficient for facilitate scientific activities, but you know, um, but I think it's important and fun. And so, what is the merit to do this for artists and scientists? And sorry for messy text, but and the most important point merit is to have that critical feedback from both sides, you know, and we can understand the different views on their own projects. And for artists, of course, sometimes they can have some technical support from scientists, and in some cases, some artistic practice leads to new scientific discovery. Because and of course, in art making or the preparation of art, they sometimes do very extreme experiments and uh, with how can I say, very strange parameter setting. In such a case, and they can find some new phenomena which is not known to even scientists. And in such a case, it can be the seed of new scientific activity, but it's it's very rare. And I do not how can I say, it's not the aim of the doing art project. So it's a merit, but I do not think it's a very the aim of the platform. But it helps sometimes. And of course, and we are doing the cyanobacterial studies, but having art project, we do some breeding the goldfish or silk moth or water lily or something like that. So anyway, so it expands the activities, and sometimes it's helpful to how can I say, expand lab skills and facilities. And of course, and we can learn some alternative styles of output. For example, for standard scientists, the normal output of the research is a scientific research article or some products or some how can I say, some applications, and do not think about the artworks as their output, but it can be an alternative output of scientific activities too, and vice versa to the artist. And for some artists, and we so the two so they need to explain their own project, the scientific project to the artist, and the scientific artistic project to scientists in such a condition that they need to uh, learn some writing and presentation skills to the different community, and it is also very helpful in some cases. And but of course there is some cultural difference between artists and the scientists, and this is very important. And for example, the styles of presentation to be evaluated is quite different. And for example, the research article by scientists versus artworks by artists are quite different style, of course. And we have a very different idea about the credit. And in science, usually at any contributors are the scientific contributors to be co-authored. But in usually in art project, as artists art and artists and supporters are highly discriminated, and usually an artist name with the technical support of from blah blah blah, and so like that. In scientific sense, the community sense, it can be the co-authored, like a collective one, but but anyway, so we have some different the custom for credit. And also the review process is quite different for selecting the ones, and in scientific research article, the still the peer reviewing is the standard, and while in art, how can I say, art award or something like that, and usually the jury is open to the public. And of course, even in the science, we have a lot of discussion about peer review. And some alternative trial is now ongoing, but and also and in science, even though the research article is declined from the editors, that we referees need to fill the very detailed report on such losers. That it's very important and very helpful to learn a lot and from such a critical comment, even though if it loses. But normally in the jury selection and the losers in the art awards, we cannot have the very feedback from the committee. Of course, some selection processes that we can have some feedback from the jury, but it's not very normal, and it's still rare, I think. And so, and of course, and for artists, and sometimes the regular lab rules in the biological lab is too stubborn or too troublesome, and also the turnover rate of the project is also sometimes quite different. And for example, speculative design is the turns many subjects quite rapidly from the sense of the experimental biologist. And of course, the turnover rate is quite different even among labs in biology or in science. So it's not only for the science art difference, but anyway, the turnover rate can cause some conflict. Okay, so to manage the such science art long-term science art communication, we need three very important words. So one is patience. The second is patience. And the third is patience. But that's a joke. And I think that there is only one demand or one important one is the tolerance to acknowledge and enjoy difference, which is the only one the need to do. And there is some additional difficulty in Japan, but this is some complaint. It's not so very important, but anyway, it's very separation of this or sectionalism is highly developed in Japan and possibly compared with European countries, but I don't know exactly, and more or less, and the situation may be similar, but anyway, but so probably you may know that the University of Tokyo, and that is our first university in Japan, was the first world first university lacking theology department, and the world first university which has the engineering department. So in 1880 or something like that. And so at that time, it was very, very advanced one. But the art or music and fine art are not included in standard universities in Japan, but only in smaller colleges, so-called art universities. So there is no agency to connect them, and so to do some assign. So there is some barrier. Okay. Okay, 10 minutes more. Okay. Sorry. Okay. And this is not so very important, so that I will quit my talk. Sorry. Thank you.
Okay, thank you very much, Hideo, for the great talk and for this different perspective on your work than the other talk before. So I think we have one question from Carolina. Carolina, do you want to unmute yourself and ask it yourself?
Yes, of course, I can ask the question. Thank you, Hideo, again for an amazing presentation. And what came to my mind listening to you is a pretty simple question, because you said that you are giving to artists a possibility to realize a project in your laboratory. But I'm curious, what is your position and your participation in this project? Are you just a tutor, or are you a regular collaborator and you are participating in the project on each stage, and you are also like a creator and during the exhibition you are also one of creators of the project? Or is like you are just giving this possibility to use the laboratory, but artists work independently and you are just observer? This is like curious for my from my perspective.
Thank you. It depends on the situation, and the three previous three ones, three topics. I was the supervisor of the three students and postdocs. And so, and in that case, and I was tightly involved in the plan making process, and some preparation of facilities to their artworks, and also two of them wrote a paper, the thesis, and I supervised it, and also I juried them, the referees, the referees. And we made some collaborative the articles as a co-authored. But this is a so in that case, I was also involved in some education of them. And um, for for example, this the April project, which is also involved with the many other artists and also some local people. In such a case, and like a project leader of the collective the activities. And as for the relationship with George, I'm not always the an artist of their project, but I'm a supporter of his project usually. But in some cases, we feel we are doing some collaboration, and yeah, but probably it better for George to explain it like this. Anyway, so it depends on the context and the situation.
Okay, thank you.
Thank you.
Yeah, I will not explain it now, but I would like to ask a question also, because this year and also last year you did run a bio art course for science students. Could you maybe please talk about this very briefly?
So how can I say? So of course, we always listen to the science student also is listen to the presentation from artists, and in such a case, sometimes they agree to the artist's opinion, but in most cases they have some different opinions, and they do not sometimes like their artistic approach. In that case, I recommend them to consider what is your favorite approach to connect the art and biology. So therefore, I started the class to facilitate them to think about the what is their own bio art project. And of course, it due to time limitation, they cannot actually make the prototype or something like that. So it's just to make a plan or sketch of the proposal. But and in this class, and initially we have some series of metaPhorest seminars to be presented to the students, and the students the how can I say write very brief reports on that, and then they will start something about for their own biological artwork, and George is very highly kindly involved in the process. Thank you.
Yeah, I think Henry is also here. So all the metaPhorest artists coming together and support it. But can you briefly say how is this course called officially and unofficially?
It's a molecular cellular biology, actually. We do not talk anything about molecular biology usually. But
because you cannot officially put an art course within your engineering university. Okay, yeah. That's amazing. Okay, I think let's leave it like this for now, and let's save some questions for later. So I would like to pass to Christian and Regina to introduce Matthias. Thank you again, Hideo.
Great.
Thank you.
Thank you, Hideo. And we're very happy to have Matthias Rillig today in the BioClub. Thank you, Matthias, for taking your time. And let me just introduce Matthias, who we know for a couple of years. Matthias Rillig is specialist in plant and soil ecology. And with this, Matthias got his professorship in 2007 at the Free University in the Institute of Biology. And so he has, as Hideo already said, a very, very large lab. On your group photo, I counted more than 50 people the other day. And Chris and me were very lucky to have already been inside the lab, and I see later on what we could participate already. And Matthias' versatile field of research, could say, is in the area of soil and plant ecology, where his research interests include the symbiosis of arbuscular mycorrhizal fungi with plants and its effects on soil aggregation. And next to the lab, for the last couple of years, Matthias is also the Dean of Biology, Chemistry, and Pharmacy at the Free University. And also since 2013, Matthias is the director of Berlin Brandenburg Institute of Advanced Biodiversity Research. Your field of research interest, Matthias, if I just only name a few: mycorrhizal symbiosis, other symbiotic connections between soil microbes, bacteria, fungi, and plants, microplastics, and plant ecologies, which tackles a field here for scientists and artists here in our group as well, and microbial biospherics, which is also like a project which has been contributed by India Mansoor, who is one of the Rillig Lab team members, who is one of 3 team members also from the Rillig Lab that are actually part of Art Laboratory Berlin's Die Wahrhafte Panke, an art science group, which is exploring the River Panke in Berlin from a hybrid approach, art and science. And just a few examples to name about the research themes in Matthias's lab. As I said, ecological synthesis, biodiversity of soil aggregation, biodiversity of and community ecology of arbuscular mycorrhizal fungi, soil biota effects on plants and plant communities, just to name a few. And the crossing, the very constructive and always inspiring crossings that we have lived with Matthias, and very recently, is that there will be a paper published by PLOS Computational Biology, 10 Simple Rules for Hosting Artists in a Scientific Lab, also together with the artist Karine Bonneval and Sasha Spatschal, the artist that he is today also with us, and also with Vera Meyer and Johannes Lehmann. And talking about the artist, I think I do not want to take anything away. Matthias, you will probably tell us more about today. But a few years ago, when Karine Bonneval, French artist, came to you, I think that was the moment for the crossroad of artists in your lab. And we are very happy also that we could bring Sasha Spatschal in your lab. In 2019, she was 1 month in your lab with her project Symbiosis in Intraflux of the Anthropocene, which is addressing societal and environmental issues in like climate crisis or plastic pollution in the context of symbiosis as a process of planetary metabolism. As I said, the Die Wahrhafte Panke is also very much overlapping with India and James and Daniel from your lab. And we were also very grateful, Matthias, that we could use the lab together with India, James, and Daniel in 2019 when we worked with students from the Reinickendorf school to get a hands-on workshops with soil at the River Panke. So thank you so very much, Matthias, to take your time today and be with us.
Wow, thanks a lot, Chris and Regina. I guess we have done a lot over the years. So I will try to share my screen, and this will demand a moment of my undivided attention. So I apologize for the silence. So hello. Thanks very much for that previous presentation. That was very impressive. And I guess I can start by saying I'm not an artist at all. And I'm also not burdened with any knowledge about arts, as Chris and Regina know. You could ask yourself, why am I even here? But I guess our lab, and myself included, we are very interested in people that are quite different from us. I guess that maybe says it best. And we are quite a few people Now I wonder if you can see my screen completely, or if there's something obscuring it at the top. There's some shit hanging over it here, but maybe it's not to worry about it. Okay, so yeah, we're not nearly as organized as the arts interaction as we've heard from Dr. Iwasaki. In fact, we didn't even know how many people we are. we only found out because we couldn't take an annual lab group picture because of the current pandemic, and so everybody had to send in their picture, which is how we found out how many we are. We are sort of at any given point about 70 people, and we are from 20 countries, including until very recently also somebody from Japan, who has just gotten a professorship at a German university actually, and we still have another Japanese postdoc. We are a bit of an unusual lab as far as science labs go, because we have very many, an unreasonable number, I would say, of postdocs. I'd say it's over 20 postdocs, and that's because, well, people don't leave, which is probably a good sign. We don't have nearly as high as artist density, and there is not this very close interaction of science and the arts. People here basically do the science. However, we've welcomed artists into the lab, and I'll talk about this a little bit. So I wanted to show you a little bit about what we do. Regina's already mentioned some of that stuff. So we do what ecologists do. We go out there and sample stuff. Actually, this is just around the block here in Berlin, but we sample also across the entire world in all kinds of different ecosystems. We do a whole range of different greenhouse experiments. This is a greenhouse experiments on the effects of microplastics in soil on plant growth and plant microbe interactions and ecosystem process rates. And actually the study of microplastics in soil started in this lab here. Yeah, this is one of our most favorite setups, which is this little cones of soil with a little plant in it that can interact with the microbial community in that soil. Sometimes the pots get a little bit bigger. This is from an experiment of about the effects of drought as one of the global change factors that are expected to worsen in this area on plant-soil interactions. And this is larger pots, and the tubes you see here, it's just water, so that they get watered at different rates automatically. And of course, we also work at the level of the plant community, so assemblages of plants of different species. This is from a recent study done in our lab, where we assembled these cute little plant community modules in pots, and then we expose a range of global change factors to these little mini ecosystems and study their responses in terms of community composition, ecosystem process rates, microbial biodiversity, and so on and so forth. And we do microcosm experiments. This year I'm showing you is a very special one, because it's the first experiment I did myself in about 15 years. And it is beautiful. May not be to you, but to me it's the most beautiful picture in the world. It's actually only about maybe 2/3 of the experiment. It is fantastic. There's a coffee cup in there. I picked the wrong picture. My apologies for that. There shouldn't be a coffee cup there. Of course, that's completely unacceptable. But this is from an experiment with many different global change factors. In fact, more global change factors than have ever been used in an experiment ever, as it turns out, namely 10. This was published in Science a little while ago. And it's going to be keeping us busy for a long time, this question of impacts of multiple factors on biological systems, from individual organisms to populations, communities, and ecosystems. And yeah, it's something that I'm immensely proud of, and please ask me about it later. Right, so give you just a feel of what we do. We do a lot of experiments that are themed broadly around global change. Global change, as many of you will know, is more than just climate change. It's more than just changes in temperature and precipitation, but it's changes in terms of, pesticide occurrence, microplastic, soil compaction, artificial light at night, elevated atmospheric CO2 concentrations, ozone, nitrogen deposition, invasive species, and the list goes on and on. It's actually, in fact, depressingly long. And this is what we study here. And well, we did a literature survey, and we found that 98% of all papers ever published on this topic only you look at one or two factors at the same time. And so this is where we really try to do something totally new, and you can't do experiments in a normal way this way, because I mean, those of you who know about experimental design will know that because of the combinatorial explosion problem, if you have more than 2 or 3 factors, the number of treatment combinations goes through the roof. In fact, if we did this experiment with 10 factors in a completely factorial experiment, as it's called in experimental design, it would have had 1,024 treatment combinations, namely 2 to the power 10. That's not doable, so we had to do something else. This gives you sort of a glimpse of one of our labs here. We have an unusual number of incubators. We have many, many, many incubators in the lab. And the reason this investment was necessary is that we actually take replication seriously. We don't take sort of a cavalier attitude towards just throw a bunch of things in one incubator and call it elevated warming or elevated temperature, because then you have not truly replicated temperature. If you wanted to do global change experiments, you needed to really independently replicate the variable that you're, or the parameter that you're examining, in our case here, temperature. So we have 20 basically internet-controlled incubators here that can run all independent temperature programs, and we have actually many more. And our favorite organisms are fungi, not bacteria. But we have, they're beautiful, eh? Yeah, so this is our favorite set of fungi. There are 30 of them. And we basically decided a couple of years ago, we picked them by just different growth rates. So they span a huge gradient in growth rates. And then we measure everything we can on this fungi. Scientifically, it's called the trait-based approach, which means just measure all kinds of stuff and see how it relates to each other and learn about the basic biology of these organisms at a very fundamental level, really. And this has been, if I had to pick an adjective, I would call it painful. Because every measurement that you do, it has to work for 30 different species and their, idiosyncratic behavior and temperament, or whatever you call it. So they all, they do things differently, and it's sometimes very difficult to get it all squared away in an experiment. Let's just put it like this. But it's also been very rewarding, and we're learning and learning more and more about fungi in general by comparing a wide range of traits among 30 species, which is really quite different from what people usually do when they pick like 2 or 3 species and compare stuff. So I think this is actually the way forward in terms of how to learn about fungi, and maybe plant people have long ago adopted this approach and have learned a lot about plants, and so we'll try to do the same with fungi. But they're also pretty. Another topic that I already mentioned that is that we study a lot and more is microplastic. So it's an environmental pollutant that's relatively new, and probably all of you have heard of it. It's plastic pieces that are smaller than 5 millimeters, and it's mostly been studied in the oceans and aquatic environments because there it's much more obvious. It's not so obvious what the effects would be in soil, and so it was very interesting for us to find out that it actually in soil interferes with the formation of aggregates. Aggregates is what you see here. This is a soil aggregate. It's the crumbs that soil consists of, basically. And I would say this is my own personal scientific hobby, how soils make crumbs. And in this case, you can see how microplastic fibers, the stuff that comes off your clothes, how that interacts with aggregates. And you can see this is after an experiment that lasted a few weeks. These fibers are built into aggregates by the general activity of soil. And then you have these plastic soil aggregate, well, fusions or whatever you want to call it, so that the fibers are just built into aggregates, and that makes them less stable. We don't know exactly why yet. Possibly because water can like flow along the hyphae of the fungi to the inside, or because these things that stick out from the aggregates, they maybe serve as sort of levers to pry things open. We don't know exactly which one of these it is. Maybe it's a combination, but it is it's relatively new, and it's interesting to us, and it's also made us think about things like the shape of pollutants, which is something that I'm not sure many people have thought about before. I certainly haven't. But one of the biggest surprises came when we looked at the effects of fragments or beads, which everybody worked with, because you can actually buy them from a catalog. When we shifted to fibers, that's when we saw all the effects. Basically, we had years of finding basically nothing when we worked with just beads. Because the beads are actually not so dissimilar from like any of these particles that you see here, right? That the soil normally consists of. But when you see something that's quite different from the normal population of sizes and shapes, then is when you see effects, in this case, microplastic. Right, so this is sort of a bit of what we do. I left out the microbial biospherics, which maybe I shouldn't have left out, because microbial biospherics connects a little bit to what Hideo Iwasaki talked about in terms of synthetic cells. This is our attempt at synthetic ecosystems. So we would like to build ecosystems from known components from the ground up and close them off completely in matter-closed energy open systems. We described this idea in a paper in PNAS, and we are working sort of to, as Regina has mentioned, India, is a postdoc in the lab, India Mansoor, to basically realize it experimentally, which is phenomenally challenging, actually. And hardly anybody does that kind of work now anymore. It used to be done in the 70s in the context of life support system engineering for space flights, and has sort of stayed in this corner of science and hasn't really ever entered the mainstream of ecology, which is shocking and surprising and sad, and we hope to change it. Yeah, so interactions with artists so far, as Ines mentioned, has been the French artist Karine Bonneval, who basically showed up in my lab and was the first contact I've had with artists in the group. So we did not actively seek her out. She just basically showed up, sort of like, can I come by? And she produced a bunch of different art, which I thought was fun, based on the interactions that we've had in the lab. You can see them. I don't know if you can see my pointer. Maybe you can see my laser pointer. This is a piece of art that she made. I think I'll show you that on another slide. I can't remember. Based on our paper on community coalescence, which is when entire communities meet rather than just species. And you can only have that in microbial communities, because microbes are small enough that pieces of the environment that move by a variety of different forces can actually lead to the encounter of entire communities and their admixture in the environment. And she made that piece of art. And this year is a piece of art that she made out of clay, I guess, for lack of a better word, where she talked about, or ceramics or something like this, where we were talking about sound. I guess, as I said, I cannot claim to understand what the what the art theory and all this behind it is, but I think Karine tries to communicate or find ways how to relate to life that's different from us, like plants or microbes or fungi. And one of those ways is sound. And so one day she asked me, well, what sound do hyphae make when they grow through soil? And I don't know. Nobody knows. But that conversation kind of triggered a more thinking about sound in soil. Turns out not so many people have thought about sound in soil, because our picture of soil is that it's dark, that it's quiet, and that communication happens by exchange of chemicals, not by exchange of anything visual because soil is dark, but also not by exchange of noises. Anyway, so we co-authored an article on that, and there was actually a feature published in Nature on that interaction between the two of us, which I thought was kind of fun. We had Sasha Spachal in the lab. Hello, she's also in the audience. And I think there was an immense joy for the lab to have hosted her here and the interactions. And I'm hoping she'll come back soon. She's also featured in that Nature article. And sorry, I did not have a picture of the two of us for some reason. So I just stole this one from your Twitter profile. And then we have, well, I have this last year. Yeah, I think the months are just kind of, yeah, it was last year. I had an encounter with Bo Zeng in the context of a film project. He was artist and residence at the Gropiusbau. And he also approached me, and then we had some conversations and made some movies. Some other interactions that we've had is with Anja Krieger, with whom we are currently producing a podcast entitled Life in the Soil. It's in English. You can find it on iTunes and Spotify and Google Podcast and SoundCloud if you search for Life in the Soil or my name. We have 3 episodes out. It's pretty awesome. Listen to it. It's not just me. It's also a whole lot of people from all over the planet. So it's kind of fun. And Anja is, I mean, what she does is also art because she takes these little recordings and weaves them together into a fantastic product. And then currently we have Sibylle Neumeier, who's also in the audience, I think. Hello. And this is an experiment for us because she's doing an online residence. So she shows up to our lab meetings. We have 2 lab meetings every week for 1.5 hours on Mondays and on Thursdays. On Mondays, we talk about a paper. And on Thursdays, we talk about our own research output on plants. And it's been, well, very enriching to have Sibylle present on those. And also Sasha and Karin attended our lab meetings. And that was actually, it was super cool to have them there. And well, yeah, this is the paper about that interaction with Karin and Sasha. And that a question that was first kind of a silly question then led to some, well, led to asking us interesting new questions that we wouldn't have asked otherwise. And this is that artwork by Karin that I mentioned. These are these sound stations that emit sound that she's recorded. Actually, she brought an anechoic chamber into the lab, which was technology unknown to us, and replayed then sound in that object. And this is this thing that was inspired by our community coalescence theory paper. And then finally, I want to show you just real quick this paper that it's accepted and will shortly come out in PLOS Computational Biology. PLOS Computational Biology, most of you will not know that journal, but it has an article collection called 10 Simple Rules with 10 simple rules about all kinds of stuff. And so we decided to write one for hosting artists in a scientific lab, and it's co-authored by the artists that we have been interacting with, Chris and Regine, and 2 colleagues of mine, Vera and Johannes, who have also been hosting artists. And yeah, we just put together a bunch of traits that we thought were interesting to consider if you are interested in hosting an artist. From my limited experience, what was interesting and useful. I think the most important thing for the scientist perspective is to be open. I think if you approached me and said, like, I'm doing something that's completely different from me, and then but I want to be in your lab, I think then yeah, my attitude is like, hell yeah. Because I mean it could be a complete failure, but it could also be totally cool. And so I think that that is the trait from the scientist's side, is to just be open. What I found really useful about the artists that we've worked with is that they avoided their their jargon that their own discipline comes with. So it was always easy to communicate with them. Everybody was mindful of time constraints. Right, and in our case, it was really easy to integrate people because the lab, for some reason, just was super enthusiastic about interacting with artists. Probably was just something new, and also some people I think have an inclination towards the arts, which is, by the way, it's the same here in Berlin. In fact, Berlin has a university of the arts. So at my university, there's no arts at all whatsoever, not even engineering. That it's the Technical University of Berlin. So it's just as what you've described. Right. So I think that's all I wanted to say. That's been interesting for us, and I would hope to host artists going forward, and maybe also philosophers and people from the humanities in general that would be different from us and that would be fun to interact with. And thanks for listening.
Thank you very much, Matthias. I think we all could learn a lot and we enjoyed it. And thank you for your insight. Yes. Yeah, I think there is, from the point of scientists, also working with artists and labs, there were so many overlapping points I saw here today with Hideo and Matthias. And I think also having experienced, maybe from a curatorial facilitator's point of view, how to bring artists in labs and then see the collaborations between scientists and artists, I think this point of being open to approach a holistic approach, I think it's very, very important. I always come back to what is a systemic when it's an interdisciplinary collaboration. This way, it's a translational skill that I think all ends might probably wanting to learn or to achieve or be open to. I was. As we said before the talk today, Chris and I would definitely like to visit metaPhorest and be in Tokyo one day, Hideo, to meet in person. But so far I can speak and be happy to have been in the lab, at Matthias's lab in person. And what we encountered there was an impressive openness from your side, Matthias, for the artists. You always say, I don't understand. I have not also the categories for artistic aesthetics. But what I really liked is that you were so open when Sasha were in the symposium, that you said that raised so many questions, and you and the other colleagues and the scientists came to Sasha and wanting to learn. And I think this is already basic, the eye level, that artists and scientists would meet on eye level. Can you, is there any questions from the audience already here to Matthias' presentation or the references he did to the artists in the lab? So far, not yet. Matthias, can you tell us, because we actually provided that, and we hope also Sasha will come back with her project quite soon to your lab with a mentioned project. I put in the chat box, I put the link to, if everyone wants to have a closer look to Sasha's project. Can you tell us a little bit how you experienced that? I mean, she presented in the colloquium and she researched and she had a lot of conversations. How was your experience exactly then?
Oh, I'm not good talking about my experiences. But well, it was just interesting to see what somebody else does, and that it is completely different in a way from what we do. I learned from Sasha about artistic research, that artists also do research. And well, there was a bunch of other things I learned about sound equipment, because she had some fantastic sound equipment. And it was just fun to talk to somebody that's I think, completely different from yourself, because, you know, I mean, Sasha called it, we quoted this in the Nature article, but outside of your bubble, or you said it somewhat more elegantly, but I mean, when I talk to people, it's always people like me, right? So I don't usually go to art exhibits. You would not find me in, you know, I've visited you, of course, but I don't usually go to art exhibits a lot, and so I don't encounter artists. I'm not part of the Berlin art scene. And so when somebody comes with an interest to me in what we do and asks questions, I find it super interesting to do this, to engage in that interaction, basically.
Mm-hmm. What I was also intrigued is that the mycorrhizal network phenomena is that there was a lot of overlapping with India and other colleagues in your lab and Sasha. And I think this was also phenomenal from my point of view, that there was this openness to both sides' knowledges, and also the embracing of artists' knowledges. So very often we have made the experience that a lot of scientists think then, okay, art works, and I think we could come back here what Hideo said at the beginning. What would artists in labs would then do out of the scientific project? Would it be actually a sort of aesthetically uprisal of what's been researched scientifically, or is it a modulation of the research knowledge, or is it actually a new setup? And I remember in the presentation of Sasha in your seminar that it was a walkable installation with experience that's possible and for a broader audience. And here I see the potential, what we call hybrid art, or just artists in general interested in biology here in our case today, is that to actually broaden the research knowledge, which was actually so close in an academic context. As international your lab is, but it is actually academically closed. And so what I see with the artistic projects as well to open up, which is a big potential. Maybe we could also come to an interesting discussion, objectivity and subjectivity. Hideo, I wanted to ask you, you were talking as a science relation to life as subjective, or an artist's bioaesthetic bioartist's relation to life as objective. Actually, we would rather say the opposite.
Well, I do not use the term objectivity as regularly or but properly as Jens do that. But so and well, and I mean that well, of course, the objectivity and subjectivity relationship is the philosophical core to think over it, what life is. So, and yeah, but I meant in my presentation that for the objectivity that and much more very standard how can I say like how can I say the science is much more objective or something like that, and so this is already a narrative, but and so the life of course the life is not an how can I say a notion which biologists are scientifically termed. So there are a lot of cultural or some historical aggregates or complex which cannot be objectively dissected in a purely scientific way. And so we, so therefore that we, but of course, the science can also open up some route, how can I say, to think over the relationship of the objectivity and subjectivity in life notions, and in one sense, and so, but here, yeah, it's quite difficult to say something about that, but anyway, and I think it's very important. Yeah, actually, so anyway, so if we feel some liveness and we acknowledge some subjectivity in the object, so so I'm always considering, and I'm curious, what is the origin of such an objectivity and subjectivity and issues in acknowledging life.
Maybe this is also part of generational, like within bioart. Certainly, I guess we could call it the second generation of bioartists who've emerged in the 2010s. Very much have this post-humanism subject matter, and we did a series around a lot of these artists. We called it non-human subjectivities, because we found that they were constantly trying to approach the subjectivity and the agency of non-human organisms, so in quite varied forms. Yeah.
And then again, but talking from the artist's point of view, there is a whole world of the term which we could call the aesthetics of care. So also caretaking in a way of decentering the human attention and actually coming from the organisms, or coming to explore and work and argue from a sort of subject matter, subjective point of view, from plants, organisms, animals, etc. So just that's adding to, I think, a conversation which would be also like phenomenological, really like a philosophical conversation indeed at that point.
I think Tuja had an interesting question for both.
I read Tuja's question.
Or is Tuja want to ask?
Tuja, do you want to?
Yeah, sure. As I write, I'm curious about the challenges of co-writing a paper with an artist from a scientist perspective. I wonder what kind of experiences you had since you are co-writing with artists. This is a question to both Matthias and Hideo, of course. I mean, it's just
right. I mean, this was sort of an article that was an opinion article that we wrote together with Karin and another colleague of mine. And basically the role of the artist in this collaboration was the source of the idea in the first place. And therefore, in terms of co-authorship rules and regulations that we've heard from Hideo Iwasaki's talk in Science, she was basically by default a co-author on that paper. And she also contributed the only illustration to the paper, which was a depiction of her artwork. And it was the first one for us. And it created all kinds of trouble because she didn't have an institutional affiliation. It's just stuff that you never think about. But when you have, yeah, we also had in the other paper, you know, they're not, they are, you know, what do you call it, freelancers. So they don't have like, it's not an institute of biology or whatever, but it created all kinds of mayhem down the road in the editorial process, like what is the affiliation of this person? It can't just be a street address in France. Well, yeah, it is. But yeah, I mean, there was, I wouldn't say so much as co-writing that paper. as it was a payment of intellectual debt to having been the origin of the idea in this case. And in the other case, that Nature paper, it was independently written in collaboration with the Nature Publishing Office. That editor worked with me and worked with Karine in that case separately to produce this article. Yeah.
Well, and so as for the I think that anyway, and for scientists, the writing paper is to producing anyway some representation of their idea and their how do you say, their performing experiments. So therefore, it's also the representation, and especially in biology and medical sciences, the figures and the illustration are the core to the published article. So, and I think that it can be reconsidered as also the visual culture harvested among the scientific community, even though they think them as artists or designers. So I think that we have several ways of representation of the world, and which can be sometimes better to be illustrated in the scientific figure or documented in the scientific article. But in some cases, for some issues, the scientific description is too stubborn. And in such a case, we need some different representation style. For example, to deal with commemoration of something like that, cannot fit to the basic biology discipline. In such a case, we need some different the representation style. So of course, there is a very strict difference between the scientific writing and artistic representation. But I think if you think about the very, how can I say, the multi-layers of life notion, we need anyways combination of different way of representation. So to me, the scientific representation or writing paper is one of them. And to make the stones or so making a paper cutting is another style of representation related with it. So and to me that's it. But how can I say? So I think so. So anyways, I think that the scientific description is very stylish and a very elegant way of representation of something. So I would like to stick to it, and I really acknowledge that power of representation. But it's so anyway, and I think that artists also can have some how can I say I think that the artists who try to write something scientific text or some explanation to the scientist can learn something, and in which they have alternative way of representation. So I think I hope it may merit for artists too. And of course, at the same time, we need to learn some alternative ways of artist way of representation of the world.
Thank you, Hideo. That also reminds me that with every art science collaboration project more, I gain the impression that it's always fine to step away from that antagonism, right? Or this dichotomic, the dichotomy, sorry, of the science artists, which is probably some more overlapping and intermingling. And I like the idea of crashing and hacking systems and then maybe asking questions in another field that you would never ask, or the people, the specialists would never ask there. So this is the giving and taking, which is really wonderful. Or when Matthias, when you speak about the artist contributing to a paper or then the affiliation, I think we probably have to hack more science papers in a way, or I guess academia, and to undermine and to undergo. I'd like to come back to some more comments, and I read Georg my questions. Georg writes, art projects sometimes have critical potentials and agencies which might not align with the university's policy. Does the freedom of research also apply to art projects?
It's a good question. I never encountered that situation, so I simply can say I have no clue. Can you give an example of what you mean?
Yeah, so thank you very much for reading the question. So some artists, they're really interested in really pushing the boundaries of what's possible and what not. So example, genetic engineering and so on, which might not reflect very well on the host institution. And I understand it might be not in the interest of the lab to host projects which would cast not a favorable light on the lab and institution. Huh? Did you ever come across projects like this?
No.
Okay.
Yeah. So, and if it had the potential to cast the lab in a negative light, I would not support it. Quite simply. Yeah.
No, not the lab, but so maybe even 20 years ago, when artists wanted to do just genetic engineering on plants, and I tried to knock on doors of the labs, and the scientists themselves were interested, but then from the universities came, no, it's not. It would be bad for our image if we would collaborate with artists on genetic engineering. But maybe things have changed, so that's very good.
All right. So yeah, no, haven't encountered that situation, and I don't know what our university would do. I mean, it's called Freie Universität. So presumably they are for freedom.
Yeah,
and definitely we are guaranteed freedom of research. I mean, nobody tells me what to do and what projects to engage in. So that's completely free. But in the case of artists, yeah, it must abide by genetic engineering rules. I mean, we are a genetically engineering certified lab. We have S1 lab status, and so we can do genetic engineering work. And we have to abide by all other rules about working with pathogens or whatever. So yeah, as long as it doesn't conflict with the rules, and as long as I would find it interesting, why not? But that hasn't occurred really, so I don't know how I would react, to be honest.
Okay, that's great to hear. Thank you.
So maybe I read a little bit about the comments. We have a really active chat forum here. And Carolina was asking, and I give this question to you both, but then I read also the comments. What do you think about paper as form of art? Maybe that would be the bridge between arts and sciences representations. It is really hard to force the art world to see writing as a visual art production. And then Mark comments, not sure if the goal is to get into the traditional science paper by hacking, more interesting to build new structures and systems. Then Kat writes, though there have been occasions where publications have been hijacked as an artist intervention. Whereas Carolina writes, yes, Mark, of course, I mean a new form, but still writing. Then again, Kat, isn't that the aim of the platform design of the Journal of Artistic Research? So here we have already a parallel discussion. I wanted just to share this with everyone. And do you want to comment on that, Hideo, Matthias?
I can maybe say one short thing. It is our observation and that of many other people that in order to, and I'm sure this is not exactly what you mean, but in order to get into more desirable journals, let's put it that way, nowadays, the demands on display items, I mean, this is art inside of the articles, the figures, the display figures, which can be, they can be conceptual figures and they are pieces of art. I have some very talented scientists that they make the illustrations and they are, to me, they are art, not in the sense, I think maybe what you, well, I don't know. To me, they're art anyway, and they're beautiful, and they serve a very important point to bring across the message of that paper. In fact, when we write a paper, I mean, I have these stacks of like sketches of stuff where I doodle around, and whenever I write a concept paper, I first I try to make an image, and then I write the paper following that image, and there are some people that can put this image into something that other people like to look at, not like what I do. And this is really that the game has really raised in the scientific publishing in my field for sure, that there is, if you don't have top-notch, state-of-the-art, excellently executed figures, you will not make it into the most desirable journals. And I think this could be, pretty sure this is not exactly where you're going, but I mean, this could be a source of a symbiosis between artists and scientists if they can somehow depict what we are trying to convey in our writing in a piece of art that could be part of the article. Then
yes,
I think
I'm
it would
sorry,
be wonderful.
just a short comment, because it would be kind of illustrating, and I think we are talking about a little bit of I mean, about something else. I meant
I know.
writing created from scratch by an artist and being presented normally in institutions like museums or galleries as a visual writing. And what you just said, Matthias, about this sketching before writing the paper is also a really typical, I mean, it's typical for scientists, and for me it's also kind of sketching. Probably it would be really interesting to present something like that in form of an exhibition. But yeah, for scientists it's usually even not sketching. This is like preparing a paper. A real work art is like the ready paper published in a good journal. So meanwhile, in gallery, I believe it would be really hard to have an exhibition consists on just papers prepared by artists. Still, the artwork is focused on something like object to be seen or interact with. So I think about this kind of field divisions, because we have this scientific area for writing and this artistic area which is for objects. Meanwhile, maybe it would be interesting to try to remove this division.
Yeah, I mean, I don't know how to do that, but I think that sometimes it's been an interesting experience in our paper in Science this year. What we had the experience was not necessarily an artist, but it was an editor in Science read and commented on our manuscript draft that was not from our field, and that person, it never happened to me before, but that person removed all jargon from our article because it was not an ecologist at all. And I think it in the end made the paper actually better because not all the jargon is needed. And so maybe it would be an idea to have the next paper written by an artist because it would be more jargon free and a bit more direct, and then would serve the purpose of communication better. Yeah.
This is actually something we have thought a lot when we from Art Laboratoire Berlin were asked from the biotechnologists from TU Berlin, from Vera Meyer, to take part in that citizen science project, Mind the Fungi. I just put it in the chat. We just published the Mind the Fungi book after this 2-year project. So there were artists and designers. We have also Farah Peluso here in the forum. And our biotechnologists were very keen actually to publish this not as a solid paper, but now we were approached by the TU University publisher. And so what we did, and the whole book is, I sent the link, is fully digitally available. And this is of course I'm biased, but you can like it or not. But what I find interesting, this is a mixture of paper and essay text and artist text and interviews all in one book. And the approach was very non-hierarchical. And we basically summarized what segments were in this 2-year projects: hands-on workshop, lab research, exhibitions, art and design works. And then in the end, what I find interesting, you find really, you might zip through if you're interested, and you might really find numerous genres of texts.
Right. There were scientific papers from two different science experiments from 2 different departments of the TU, and then also point of view of the 2 artists in residence. There were our curatorial essay, and then also a report from a citizen science project connected with the project.
I'd like to repeat, Mark wrote something in the chat, which I'd like to read. Both speakers shared a single slide with an overview of best practice challenges. Could you, Matthias and Hideo, share it as an image? Mark would like to put it on the forum for further discussion. So maybe this is also...
I think I can share that one once the paper is published, because it's published open access then. And it has a link that figure itself has a DOI then.
Great. In the chat or outside the chat, have I forgot a question?
Also raise your hand if you want.
Please, or unmute yourself.
I think somebody asked Henry if any of my students have presented their thesis as an art project, and the answer is no. I'm not sure we could do that because it would be a Master of Science, and I think university regulations would not allow presentation of an art project as something fulfilling thesis requirements.
Can I ask additional question regarding what Matthias said a second before about these regulations? Because this is usually really curious how you are able as a scientist to invite artists being not educated in biology and being even not after a course of biosafety protocols, etc. How you are dealing with that? Did you have maybe any troubles with the situation that someone asks you, why do you have this person in your laboratory because the person is not a scientist? Because I know also that usually in my case it was like that. It's like doing it without asking certain organs and officials because it could be maybe more complicated doing it like that, like officially asking and trying to write a paper of agreement or something. So yes, especially you both having already few projects, probably you have experiences with that.
Well, I can say something real quick, and that is Karine had her own lab. Actually, she used our lab only to a minimal degree. And everybody that works in our lab will have a mandatory safety instruction, just like what Dr. Iwasaki said for his lab. It's any person can work in our lab as long as they have the safety instruction. So I think that's not a large concern. And actually, all of the artists had pretty good biology background, which was also exceptionally helpful.
Jens, you had a question.
Yeah, I found it very interesting in Hideo's talk that he mentioned that there's also this attempt to provide bio art classes to scientists with a different background. And on the other side, that Matthias also raised the question, well, there's a university of arts, knowledge production and research is so segmented, right? And what would happen if you put a scientist in an artistic context? Why not having a scientist in residency? And what does this mean, right? So just turning it upside down and to turn this totally normally considered situation of fruitful misunderstanding when both worlds collide into something else where you put the situation upside down. So there is maybe something, and there's also an aspect in Hideo's list that I found interesting, that you mentioned conservation, right? So typically, I think the only scientists that only inhabited regularly art institutions are conservators. They're in the conservation departments, they're chemists, they do research on biorestoration, on bioremediation with Pseudomonas bacteria to clean paintings from patina, and they analyze pigments, they restore pigments. So they have natural scientists in art organization. But to conserve, so now if you conserve bioart, it's totally upside down. It's to use conservational methods to conserve what is alive and not to conserve aliveness to what is already dead. So it turns the question upside down again. So it would be very interesting in how far we can consider that conservation is maybe a very interesting practice to bring art and science together. So I would like to hear both your reactions to that.
Yeah, exactly, and I completely agree to you, and that actually, so we are now starting the collaboration with the art university in terms of the possible conservation of the biomedia art, and we actually had some it's some granted project, and now we are thinking about which type of conservation is possible, or what the conservation and restoration and mean to biomedia art or something like that. And actually, and in that case, I often goes to the art university. Likewise, actually, the scientists in residence in the Tokyo University of the Arts. And also, I understand your suggestion, and yes, and I think it's very interesting and important to consider the further for this field. Yeah. Thank you.
Yeah, I think it's a great idea. Why not? We've talked about it before, like the scientist in residence in an artist shop, and that colleague of mine is also co-author on a paper, Johannes Lehmann from Cornell. He's trying to do something like this, I guess, a little bit, and he's also said that we as scientists can learn from artists how to fail. I'm not sure exactly how he meant, but he he said that I'm not an artist, so I don't know, but he said artists try many different things and are very positive about when things don't turn out the way it does, whereas failure in our case is looked down upon very severely, right? I mean, oh, your experiment didn't work, that's a catastrophe. And so, I mean, there may be things you can learn about how you go about your daily business from artists that maybe what we do is not the most productive way of doing it. I mean, in terms of just in behavioral patterns or how you discuss and how you critique things. I think some of the cultural aspects, I think they could be quite interesting to import and incorporate into what we do in a science lab. And of course, the cost of that would be productivity, right? I mean, Hideo has also mentioned that there's a productivity cost if you, at least in the short term, maybe in the long term, there is a creativity bonus, but in the short term, there is a productivity cost. And I guess it will be for people to individually decide if they're willing to incur that cost or not. I wonder how many of my colleagues actually would.
I know of one case in Waseda University in Yogyakarta, Indonesia. Professor Irfan Piajambada has his undergrads actually are assigned to make a bio artwork. So they actually have scientists in training basically have to go and work through the eye of the artist as part of their training. I think Kat also wants to add something. Let me just see.
Yeah, I do. So my studio is hosting 2 scientists in residence through the Studiotopia program, which launched last September. Yeah, so Studiotopia is this H2020 funded project across, I think, 13 European partners who bring in artists to, 7 European partners, 13 artists who then host scientists at their studios for a period of 18 months. And it's not the first scientist in residence program by any means. I mean, I know that there's been long-standing scientists in residence at the Slade at UCL for quite a while. But what's interesting about Studiotopia is that they're also doing meta research on how there's a difference in the kind of relations that emerge when you shift this power hierarchy a little bit by introducing the scientists into the artist practice rather than the other way around. So that's ongoing until the end of next year. I have, as I say, 2 scientists in residence, and at the moment, we're working very collaboratively on kind of developing new concepts and ideas that are a synthesis of all of our work. I mean, I would say that that is somewhat more collaborative than the experience I've had as an artist in residence in scientific institutes, where the impetus has been mostly upon me to bring an idea and then people kind of adding advice to help shape it. But that's just my personal experience of that. But what I think is really interesting through lots of my experience of working with scientists is that you do see a reciprocal kind of changing of perspective if you have deep enough interactions with people from another field. And this is my experience working between art and science. I mean, I think this happens in a lot of interdisciplinarity in circumstances, interdisciplinary circumstances. But I think while there are lots of opportunities for kind of more straight interactions where you have, aesthetic aspects coming from the scientist, from the artist, and factual aspects coming from the scientist, I think really the most potential for interdisciplinary practices to make a difference is when you have deep conceptual engagement from both parties, which is going to transform not only the concepts that come out and produce a different kind of knowledge than could come from the other disciplines, but it's also going to foment change within the actors themselves. And that's something that I think there's lots of potential to look into from a meta perspective as well, like what kind of transformation, particularly what type of long-term transformation happens in the actors. And this is the problem that's been faced by people looking into interdisciplinarity for decades now. How do you gauge what the long-term transformations are when you have these collaborations or residencies in between art and science?
Can I ask something? I mean, what do the scientists do?
My scientists for Studiotopia. One looks at the paleological data, big data records, and looks at how you can do machine learning across those, across that data to understand kind of important tipping points in evolution. And the other one works with slow processes of wastewater treatment and preservation of ecological, like wetlands and water processes that goes through the earth, basically. So earth and hydrology.
Okay, so the first one works with data, and the second one needs a lab. So how does that work?
Well, he has a lab, of course, at his own institution.
So how does the residence then manifest itself in practical terms? I mean, I imagine he hangs out in your studio or something.
We've had some issues with being together physically, of course, because it launched during Corona. Ideally, what would have happened would be that they would come and work with me in the studio, understanding what my artistic research processes are, and then seeing how that affects their own research, and us coming together collaboratively to create something that either leverages only my artistic research methods or brings in their research affordances from their own institutions. Now we're doing that remotely, so we meet and have those discussions and share our methods, and are coming up with a concept now to work on.
Thank you, Kat, for your contribution. I'm so grateful that you are here with us today, Kat, but also that you are coming from all sides. You are coming from science and art, and you worked in science journalism, and you are now mainly working as an artist. So I think you have best perspective, and you are also really hacking disciplines and institutions. And you're also a really dear member of DIY Hack the Punk. And my question would be, to your experience, Kat, to just break these hierarchies and to break these dichotomies. I cannot repeat myself enough to have these opposition samples, like analytic, irrational, objective, subject. It's all not that, obviously. So what was surprising points for you to actually come against these traditional antagonistic opposition partners? Well,
yeah, it's interesting, and the way that I approach it, I would say, is only one way to approach it. But I became fascinated very early on about how people judge other people and the worth of their work. And this is something that I don't talk about very much, but it's something that I have been very interested in and aware of since I trained as a chemist, years ago, and something that I learned a lot about going through all the institutions that I have been through. I mean, I've had, as you say, a very varied background, and I've been invited into a lot of very interesting places, most of which wouldn't have happened if I hadn't have made quite a study of how one carries weight in different institutions. And I make a conscious decision normally which side of myself needs to be shown at what point and how to present that. And I would say, I mean, I do it less now, I would say, than I used to, but I basically hack my way into institutions, and I deserve to be there, but at the same time, I don't reveal all of myself to these places that I work with. And it's interesting. The world has changed very much since the early 2000s when I started doing this kind of thing. And now it's not as necessary to not present the whole unified kind of interdisciplinary perspective. But before, you know, and in some cases, it's still the case that if you present yourself as having experience in multiple worlds, you can actually do yourself a disservice to whichever one you're talking to at the time. So it's very much on an ad hoc basis. One has to work out how one is best going to be presented, and which things to reveal and which not, in my experience. But the most time-consuming and difficult of the tasks involved is working out how to move between structures within institutions and to move around structures and institutions and funding in order to get the work done that you think is necessary and important to do. And I haven't always succeeded at that, but sometimes I have, and it's been very worthwhile when that's happened. Yeah, but as with successful collaborations, I think the key is finding people that really understand the same perspective, who are in key positions, and staying in contact with them, despite the kind of structural problems that have arisen through incremental refination of the way that we do research and the way that we set up institutions and funding bodies. I mean, we all work within a long legacy, starting in the Enlightenment, of hyper-specialization. And the work that we're all doing now is trying to combat that somewhat. But I think there's still, I mean, we hear that there's still a long way to go in a lot of places.
That's a great final statement. It's still.
I think one of the next sessions we should have, Kate. I really would like to hear you more from you and about this. So
Are there any more comments or questions? Questions we have probably numerous, but maybe for today. We have also a Facebook chat group. We have many ways to reach out to one another. If anyone is here new or wants to get in contact with anyone, also let us know.
Just contact me or Regina or Georg, and we can connect you in the circle. And Mark says something. Okay, and
Mark agrees with Kat's hack approach, maybe like man in the middle attack in hacker jargon,
or what I call the role of intermediaries. As Mark
the intermediaries are.
There's a bunch of research about people like that called interaction experts, but it doesn't seem to be very well known, but that's another term for it.
Okay. Hideo, Matthias, we want to thank you a lot for today, for all you've offered us. From our part, Art Laboratory Berlin, we want to thank the Berlin Senate for supporting us these last 2 years to do some special projects, and also this and the last Viral Cloud were also featured in the Vorspiel program of the Transmediale and CTM. I think Mark and Tengel had asked what's up next. We don't quite know. I think Georg and us will speak about that in the next days, but the Viral Cloud will continue. I think we need maybe a 2-week or 3-week break, but around the turn of the end of February, beginning of March, I suspect we'll have something going.
But if anybody wants to step forward, please get in touch with us and we'll set it up. Yeah.
Okay.
Okay. Thank you so much, and stay healthy. See you soon. Thank you.
Thank you very much.
Thanks. Bye-bye.